Yes, we are live. You can start now.
Okay. Am I audible?
Yes, we are live now. So welcome, Dr. Shikdar. Uh, this is a webinar series that has been organized by our department. You know that. And over to you, Momita. Momita, ma'am. Dr. Momita Das will moderate the whole session.
Yes. Am I audible?
Yes, Momita, you are audible.
Okay. Thank you, sir. A very warm good evening to all of you. I welcome all the participants to the second day of the International Webinar Series organized by the Department of Applied Nutrition and Dietetics, Sister Nivedita University. And this webinar series, as it is entitled Unravelling the World of Nutrition. So today we have with us Dr. Kunal Shikdar, a very eminent personality, and who is going to help us peep into the energetics and genetics of a failing heart. Now, before we move on to Dr. Shikdar's talk, I would like Dr. Nilanjan Das to introduce him to all of us.
Thank you, Momita. First of all, a very good evening to all the respected dignitaries, all the participants, students, and my fellow colleagues. It's a pleasure for me to welcome Dr. Kunal Shikdar. He is a friend, old lab mate. Together we have done a lot of works. And I'm just telling a little bit brief about Dr. Kunal so that the audience can understand. So Dr. Kunal has done his MSc and PhD from Department of Physiology, University of Calcutta. He is also a former postdoc from Department of Medicine, Thomas Jefferson University, Philadelphia, USA. He was also assistant professor from Tripura Institute of Paramedical Sciences. So currently he is holding a position as assistant professor in the Department of Medical Biotechnology and Department of Sports Science, Cardiology and Metabolic Disorder, Jivon Division. From Ramakrishna Mission and Vivekananda Education and Research Institute from West Bengal, India. So Kunal has a lot of research interest companies from different areas. Especially, he is now focusing on several aspects of cardiometabolic disorder from the different aspects of cardiac health, also lipid metabolism, non-alcoholic fatty liver disease, and several intercellular crosstalks during the development of heart failure. So to that end, today we are going to hear from him one of the very interesting topic, that is the peeping into the energetics and genetics of a failing heart. So over to you, Kunal. A very warm welcome from our Department of Applied Nutrition and Dietetics, Sister Nivedita University. So please.
Thank you, Nilanjanga. Thank you. Thank you all for giving me this platform to talk about my research work. And I must express my most humble gratitude to VC sir and also the faculties of the department, especially Dr. Nilanjan Das and also Dr. Tom Shubro. So basically, I'm working in the field of cardiology. I'm a cardiac scientist right now. So I'm associated with Ramakrishna Mission University, Vivekananda University. And I'm affiliated with two departments, basically. One is the medical biotechnology, and then also there is sports science department. And I'm a scientist in the Ramakrishna Mission Hospital. So we have like a very big unit of research. It's called Jivan. Jivan Research Unit. It comprises like over 30 scientists, like the clinicians and also the basic scientists from different fields. And we have like 5 subgroups working in different areas of biomedical research. So our group is called the Cardiology and Metabolic Disorder Group. We have 3 basic scientists and 3 clinicians. And also there are other 4 groups. One is like neurophysiology, and then there is another group, cancer biology. And the 4th one is the microbiology group, and we also have a very good group of statisticians. So altogether, we are 5 groups working together and working in different campuses, obviously. Some of them are working in the Narendrapur campus, and then we have the Belur campus and also the Sherapudishan Hospital. So let us talk about some of the popular area in cardiovascular field, and also after that. So my presentation is basically divided into two parts. The first part is mostly for the students to understand what is heart and how the pathology of the heart occurs in different conditions and what are the different pathological conditions are there. And then the second part will focus a little bit on my specific research area. So right now I'm presenting my screen. Just let me know if it is visible. So the thing is that somehow my MacBook is not responding to, there's some kind of privacy issue. Let me try.
Any problem?
Yes, there's a problem sharing my screen because MacBook, there's a privacy issue, and it says that you got to open the preference and the security privacy to grant access. It never happened with, never happened before. I don't know. Yeah, I think it will work now. Just give me a second.
Professor Thakur Chaudhary, if I may suggest, if he can forward the presentation to
that is another option. Yeah, that is another option.
If you can forward the presentation, Dr. Shikdar, to Professor Thakur Chaudhary or maybe somebody else, or maybe Professor that, then you can.
This is working right now,
Okay.
right?
Yes.
Yes, it is coming.
Okay, fantastic. Just hold on. Yeah, perfect. So the title is it's a kind of interesting one: peeping into the energetics and genetics of failing heart. And this is basically a picture of myofibroblast that I stained before with two different cytoskeletal protein, the vimentin with the red and the alpha smooth muscle actin with the green, and obviously the nuclear stain with the DAPI. So let me talk from the very basic aspect. What is heart? It's a muscle, and obviously it's one of the strongest internal muscle of our body, and which is the size of your fist, located between the breastbone and the spine, just to the left of the midline inside your rib cage. And our body, the heart actually supplies the oxygen and blood throughout the body as it pumps the blood to the arteries, to the different tissues. So basically, so basically it has four chambers, two upper chambers called atria, and the two lower chambers called ventricles. The left ventricular wall is thicker to be able to pump with more force. So generally, whatever happens, if any kind of pathology happens in heart, it mostly affects the left ventricle in 90% of the cases. Since the left ventricle has the responsibility to pump out all the blood to the body. So there are like few valves present in the heart that actually prevents the backflow of the heart. This is just like a door that only opens in one way. So there are major 4 valves are present, 4 major categories: tricuspid valve, pulmonary valve, mitral or bicuspid valve, and aortic valve. So they are very much strong and flexible. They only allow, as I mentioned, the blood to flow in one direction. Now there are some interesting facts about the human heart. Like your heart beats over 100,000 times per day, and it pumps out in the process about 2,000 gallons of blood every day. There are 60,000 miles of blood vessels present in our body. That is enough to go around the world twice, which is unimaginable. And other than the cornea, every cell in the human body gets blood from the heart. The average heartbeat of a woman is interestingly about 8 beats faster than a man's heartbeat. And more heart attacks generally happen on a Monday than any other day of the week. Laughing obviously is a very good therapy for heart, as it reduces the stress and gives a boost to your immune system. So how does a heart attack occur? Heart attack is a sudden severe blockage in a coronary artery caused by plaque and rupture, and that forms the blood clots. Coronary artery spasm is an involuntary closing of the walls of the artery that usually occur for a short period of time, but can cause severe blockage. Now, so what is heart disease? In a nutshell, any cardiovascular disease or CVD is a very serious health condition that keeps the heart or blood vessels from working properly. And although some people are born with certain kind of cardiovascular disease known as congenital heart disease, but that is a very few, a very small number. But most of the people, they actually develop the cardiovascular disease in their adulthood, and mostly due to some kind of lifestyle habit or lack of exercise, etc. So it is always preferred that to eat a heart healthy diet. And what is heart healthy diet? It is nothing very special. It should contain a lot of vegetables, antioxidants, fresh fruits, and definitely a very less amount of saturated fat. So starting a good habits with the food that you eat early in life is a very important way to help keep our hearts and bodies healthy for the rest of our lives. So this is a very important level that we often see in, so when you get any kind of food packet, right? Or like energy drink, something like that. So it always comes with this kind of level. So you need to read this level very well. This is mostly for the students, that it starts from here, the number 1, which is the serving size. You need to see what is the serving size, how much you need to get. Like if you are having some kind of protein drink, like whey protein or something like that, you cannot take a lot, or you may not take a very less amount. It will not work. So the proper serving size will be written over here. And then the serving per container is also there. And then check for the total calories, like how much calorie it has. And then the saturated fat, trans fat, and sodium. These 3 things are not good. I mean, as you are all dietitian, you are studying about nutrition and dietetics, you know this very well, that saturated fat, trans fat, and sodium, these things you need to limit in your diet, and get enough of these nutrients like vitamin, different kind of vitamins, calcium, iron. Although too much iron is also not good, but at least you need to have some. And then a quick guide for the percentage daily value, like how much percentage the daily value is. It should be 5% or less that is low, and 20% or more is high. So it should be in between that. So as I said before, that eat plenty of fruits and vegetables. Try to eat a variety of fruits and vegetables, like different colors, like you can have green and orange and yellow and red, whatever you like, you need to mix and match. And then obviously the whole grain foods like bread and oatmeal and brown rice, and eat fish, because fish has a lot of omega fatty acids, omega-3 and omega-6, very good combination, especially the sea fish like the salmon and trout and herring. And obviously limit your intake of saturated and trans fats, and limit the amount of red meat you eat, because red meat also contains a lot of saturated fat and triglycerides. So now you can plan like that. Write down everything you eat and drink for a few days, identify your good habits and what are the bad habits, like what kind of bad snack, and drink you're having, like sugary drinks and all. And what is one unhealthy eating habit you can swap for a healthy habit. Like if you like to drink something in the morning and you always choose for some kind of soda or Coke, something like that, you can change it to a fruit juice. Maybe you can add some salt or some other flavors to make it more, refreshing, but you need to change that habit. And obviously during grocery shopping, ask your parents or guardian that if they can help. Now, coming to the energy part, energy, as we all know that it is required for every organ, every tissue, every cell of our body. So energy is a kind of a double-edged sword. Too much energy is bad, and too low energy is definitely not good for our body. So you need to limit your energy intake. If you gain weight, that might mean that you took in more calories than you needed. And if you eat fewer calories than you use, you can lose weight. So it is very important to keep this balance. It is also important to participate in regular physical activity to help keep a very healthy weight. Generally 60 minutes, 30 to 60 minutes exercise per day is prescribed. And remember, keeping a healthy weight is best achieved over time and with a decision to keep yourself healthy. It is not like a single day decision. You cannot make quick change like within say 7 days or 1 month. It should be a long process and continuous process. So the next important thing is the high blood pressure. So hypertension is one of the most important risk factors for any kind of heart disease. We know this. So people with high blood pressure are more likely to have a heart attack or stroke than people with normal blood pressure. So you can make healthy choices that will help you control your blood pressure, like keeping your weight healthy, eating a heart healthy diet, as I have mentioned early, and getting enough regular physical activity. The next important thing is the total cholesterol. So you also need to keep your total cholesterol under control, because cholesterol, too much cholesterol, they combine always with protein or other substances in your body to form the lipoprotein, and they ultimately form the plaque, which is a thick hard deposit that can narrow the arteries. And if the arteries they get narrowed down, then there is every possibility of breaking the blood, and that forms actually blood clot, the thrombosis, and that is responsible for a majority of heart disease and stroke. Next is the blood sugar. So a very important factor. Always keep your blood sugar under control. So when you consume food and beverage that contain carbohydrates, the digestive system always breaks down into monosaccharides to get energy. But food like whole grain, fruits and vegetables, they contain a lot of fiber, and they takes time for digestion. So the absorption from these foods will be much less. And so that the delivery of the blood sugar to your bloodstream will also be limited. But if your blood sugar goes high too often, it can overwork the body's ability to keep your blood sugar in healthy ranges. And this, if it is continued for a longer period of time, it causes diabetes. So how do I know that if my heart is healthy? So whatever I talked so far, everything in a nutshell, the life's simple 7 factors you need to consider. Avoid smoking using any kind of tobacco products. Be physically active every day, every single day. Eat heart healthy diet, keep a healthy weight, and keep your blood pressure healthy, cholesterol healthy, and also the blood sugar healthy. So these 7 factors are very, very important, to keep your heart healthy. So that was the first part of my talk. It was more like a popular talk. Now we can talk a little bit about the pathophysiological changes that is happening in human heart, in a disease heart. So we generally work with the cardiomyopathy. There are several kinds of heart disease. But what is cardiomyopathy? This is one of the most common kind of heart disease. According to WHO classification, there are several kind of cardiomyopathy. There is dilated cardiomyopathy. This is in the left, the picture A. This is a normal heart. You can see all the chambers, including the ventricle and auricle, all looks normal. And then there is dilated one. You can see the chamber in the ventricle especially, it becomes very large and the wall becomes thinner. And then there is hypertrophic, the opposite happens. The wall becomes excessively thick. This is unnatural, and the lumen becomes thinner. And there are several other types like restrictive, arrhythmogenic, and some unclassified ones. So the interesting thing is that whatever classification I have described here, still about 50% of all cardiomyopathies, specifically the dilated cardiomyopathies, they do not have any known etiology. Hence, they are known as IDCM, idiopathic dilated cardiomyopathy. So right now we are actually working on the IDCM. We have a couple of projects. We are working on IDCM. That suppose a person, he doesn't have any diabetes, any valvular disorder, nothing. He's totally fit, absolutely fit. Just like what happened with Saurav Ganguly. We all know suddenly he got sick. And doctors found that his heart is dilated. But why? He's a sportsman all throughout his life. He did not have any kind of abnormality before. So these things are very dangerous. These are called IDCM or idiopathic dilated cardiomyopathy. And the reasons are still unknown. But there are some genetic predisposition and maybe some autoimmune, factors are involved there that we are trying to find right now. So these are like 2 hearts. One is the regular one, and then there is a dilated one. As you can see, that mostly the left side of the heart that I mentioned before, mostly the left ventricle, it becomes swollen up. So this is the dilation, typical dilation of the heart, and the muscle becomes very narrow. So this is a typical dilated heart. Though this is a cartoon picture, but this is exactly what happens in the body. And there are like a lot of cell populations to consider. Not only cardiomyocytes are there in the heart, there are at least 5 to 7 types of cells are there in the heart. There are myocytes, there are fibroblasts, very important cell. There are endothelial cells, epithelial cells, there are nerve fiber, and also there are a lot of immune cells. So when we consider heart, most of the people, they only consider the cardiomyocytes and they see what is happening inside the cardiomyocytes. Is there any changes happening or all? But this is not the right approach, because in a disease heart, what happens? Most of this cell population, they actually cross-talk with each other and they form the whole pathology. A very interesting finding was published in Circulation Research. I think it was in 2011, 14, I think, that described that in a disease heart, since the cardiomyocytes, they do not proliferate well, they do not have that ability like any other epithelial cells or endothelial cells. What happens is that the cardiac fibroblasts, they can transform themselves, they can reprogram themselves to form the cardiomyocytes. So this is absolutely amazing thing that the cardiac fibroblasts, when the heart is in danger, they convert themselves to make them to cardiac myocytes so that heart can still survive and it still can beat on its normal. So all these cell population we have to consider. Actually, the interesting thing is that maybe the volume of the cardiomyocyte is the highest if you consider a heart muscle, but the number of cells, if you consider the number, it is the endothelial cells. So endothelial cells, the number of endothelial cells is maximum in the heart in comparison to all of the cells. So what is dilated cardiomyopathy? It is a dilation and impaired contraction of the ventricles, either left ventricle or both the ventricles. So how to know if the ventricles are dilated and the chambers are large and the muscles become narrow? There is a technique called echocardiography. I will show in the next slide a couple of pictures. So we have clinicians, we have cardiac surgeon actually, and other cardiologists. They actually do this for us. They, whenever we get a patient, heart patient, they first do the angiography and followed by the echocardiography to see what is the condition of the heart. Once they confirm, yes, this patient is a DCM patient, then only we take the blood and we start working on them. So the reduced systolic function of the left ventricle or both the ventricles, as I said, sometimes it is biventricular or monoventricular. Characterized by myocyte damage, there are a lot of myocyte apoptosis happen in a dilated heart, and multiple etiologies are there. There may be one etiology, maybe 3-4 etiologies are together, maybe there is no etiology. So in world, about 31% of global mortality occurs due to DCM. So from now on, I will term the dilated cardiomyopathy as DCM. So over 18 million people dies globally only due to the cardiovascular disease, of which the major cause is DCM. And 30 to 50% of DCM is also due to familial disease. So that means there is some kind of genetic predisposition is there. So this is an M-mode echocardiography picture. I don't know if you know about the, I'm talking about the students. If, I don't know if you have any kind of knowledge about the echocardiography, but in echocardiography we basically have 2 different modes. One is called the B mode, where you get the 3-dimensional picture of the heart. And when you get a 3-dimensional picture, when you select a particular surface, a large area of the heart, then you go inside of the heart, and that is called the M mode of the picture, M mode of the echocardiography. So basically the B mode is 3-dimensional one, but you need to go inside the heart by slicing the heart with the ultrasound, and that is the M mode picture. So on the right hand side, this is a regular heart. I will explain a little bit. So this is the muscle layer on the bottom and also on the top. This is the muscle of the ventricle. And the inside part, this is the lumen part. Okay, so this is a regular heart where you can see the muscle is very strong and very consistent. And the lumen is not that much. It is kind of thin, and also it is consistent throughout. And this small area, this is a small axis. It represents the systole, and the long axis, this is a long axis. It represents the diastole, the ventricular diastole. So we can measure all these parameters to see what is happening. And this is a typical dilated heart. You can see the difference clearly. The muscle becomes very thin and the chamber becomes dilated. So one of the most important etiology that I will discuss today about the diabetes, how diabetes actually influence the heart, and it creates the DCM. So the estimated number of people, according to WHO global report, in 2015 it was 415 million, and with their prediction by 2040, within 25 years, it will be 642 million. So about a 55% increase. So prevalence of heart failure in general population is about 2 to 3% in less than 70 years age group. And over 70 years age group, it is about 10 to 20%. And the cardiomyopathy in diabetic, so that means the people who have diabetics, are they going to have the cardiomyopathy in future? It is 12% in less than 64 years age group, and about 22% greater than 64 years. So the diabetic patients are basically 2 to 4 times more prone to have heart disease or stroke. And more than 68% of people age 65 years or more with diabetes die from some kind of heart disease. The AHA, that is the American Heart Association, they consider diabetes to be one of the 7 major controllable risk factors for cardiovascular disease. So this is a very important factor regarding the heart disease and DCM especially. Now, characteristics of diabetic cardiomyopathy. So now we are looking specifically into the diabetic cardiomyopathy. So this is still DCM. So this is still dilated cardiomyopathy, but now we have we are focusing on one particular etiology, that is diabetes. So the characteristics of diabetic cardiomyopathy are myocardial dilation, just like any other dilated cardiomyopathy, diastolic dysfunction and systolic dysfunction both. And sometimes there is LV hypertrophy happens, myocardial inflammation, cardiomyocyte apoptosis, and cardiac fibrosis is a very common feature of diabetic heart. If you see a diabetic heart, I actually worked with a human heart also when I was in US. So we got the human heart from the dead patient. So if you see the heart, you can actually see the thick, the yellowish fiber that actually represents the cardiac fibrosis. It is very prominent. And then finally leading to heart failure. So now there are some metabolic switch that happens in diabetic heart. Actually, heart is a very intelligent organ. So whenever it is in crisis, it tries to accommodate itself in a better way. So the cardiac structure and functional changes, they result from a metabolic shift. So in hyperlipidemic condition environment, suppose we are talking about a type 2 diabetes, right? Who is also an obese patient. So that means he has hyperlipidemia. So in hyperlipidemic environment, the glucose uptake is actually impaired because of the a decrease in the GLUT4 and GLUT1 receptor. We know these receptors are very important for glucose uptake. So there is a down regulation of this. And to compensate for the decrease in glucose, because heart needs energy every single second, continuous, because it is beating 24 hours, right? So to compensate that, the fatty acid metabolism becomes the major conduit of energy supply. And as a vicious cycle, as the increase in myocardial fatty acid intake becomes larger and larger, the ability to use the glucose also becomes lesser and lesser. So this is the metabolic shift I'm talking about. And this kind of cardiac metabolic inflexibility is actually happening in the heart. So dependence of heart muscle on fatty acid oxidation over glucose oxidation leads to hyperglycemia and also insulin resistance. Because in diabetes, especially in type 2 diabetes, there is insulin resistance. You all know that, right? The insulin is there, but the body is unable to utilize it. So studies have shown that during the development of type 2 diabetes and also in diabetic DBDB mice is a specific model for diabetes where the leptin receptor gene is knocked out and there is a decrease in glucose oxidation and increase in the reliance of fatty acids. Now the questions are, so what kind of metabolic switch and what are the molecules or what are the molecular mechanisms and pathways rather, they are involved in this kind of switching. So when you work with any kind of such problem, some pathological problem, we usually have a model for that, right? So we generally have 3 model systems. One is a cell culture, and then there is the animal model. We generally prefer the mouse model because mouse is easy to handle. They breed faster and also the genetic similarity is much more with the human being. And then there is the patient sample, either the blood or the tissue sample. So what kind of diabetic model is there for mouse and how the diabetes can be induced? So the top one is the type 1 diabetes model and the bottom one is the type 2 diabetes model. So in type 1 diabetes model, we actually inject STZ. It is called the streptozotocin. It's a drug that actually selectively destroys the pancreatic beta cells. So that actually blocks the insulin secretion and the mice, they become diabetic after 8 weeks of the last dose of injection. I mean, after 2 weeks, they started to become diabetic, and after 8 weeks, they fully become diabetic. We actually check the blood sugar from time to time, every week, to see the blood sugar. If it becomes like over 300 or something, then we consider it as diabetic. And then at 8 weeks, we do the echocardiography of the heart. We can also do echocardiography in mouse, just like human. The instrument is smaller and all, but the thing is the same. And then we collect the blood and sacrifice the mouse. So this is the type 1 diabetic model. And for type 2 diabetic model, as you can see, this is on the right top. This mouse is the obese mouse, because it become diabetic with a 60% high fat diet. So in regular mouse, we give the regular chow, but for the mouse, for the diabetic model, we give them 60% high fat diet. No injection, nothing, just the diet is changed to 60% high fat diet. And then we have seen that the symptoms of diabetes, sometimes it started around here, about 4 weeks. About 8 weeks is also a good time point, but generally we take the 16-week tissue, 16 weeks time point. This is a very positive time point where we find all the parameters positive for diabetes. So at this point, we do the echocardiography, and we also sacrifice and collect blood and tissue. Now from that, we did, at first we did actually a microarray analysis of a diabetic heart to see like which genes are upregulating and which genes are downregulating. This is a very tedious process, actually, very long process. It takes time. And we know that a human body contains thousands of genes. So to cut down to a narrow margin is not an easy work. Anyway, so from our sample, we found that HNG-CS2, so this is one enzyme which is responsible for ketogenesis, the rate limiting enzyme for ketone body production. And then PDK4, this is another enzyme, which is basically an enzyme of pyruvate dehydrogenase complex that actually converts the pyruvate to acetyl-CoA, and that finally converts to beta-hydroxybutyrate or the ketone bodies through the HNG-CS2 enzyme. So these two candidates are highly expressed in the diabetic heart, and we did a pathway analysis to see that these pathways are actually involved, the AKT phosphorylation pathway and ARC pathway and all. So next, what we did, so since we found that the ketogenesis is responsible, at that point of time, there was kind of confusion, because ketone body is not always bad. It can also be utilized for energy production and all. But we found some paper where they have shown that a ketone body can also cause the apoptosis of cardiomyocytes and cells and other cells if it is produced in a very larger amount than usual. So what we did is that first we tested these enzymes in our from our heart tissue, the left ventricular heart tissue. So this is just to explain that chow means this is normal diet fed mouse, and the HFD means the high fat diet mouse. So we have 2 and 2 samples, the representative samples for both these groups, and we have seen the expression of the protein HMG-CS2 and BDH1 and PTK4, and all these proteins are upregulating, as you can see, in high fat diet group in compared to the chow or normal fed group. And we also did the real-time PCR over here to see the mRNA expression. mRNA expressions are also increasing in a similar manner. But still, we are kind of confused that if the beta-hydroxybutyrate or the ketone bodies are that much dangerous for the heart. So what we did is that we isolated the neonatal cardiomyocytes from the neonate's heart, the mice neonates, the mice pups. And then we treated these cells with the beta-hydroxybutyrate in different concentration, like 1 millimolar, 10 millimolar, and 100 millimolar. The physiological condition, just to mention, it is in between the 1 to 10 millimolar. And we have seen enough apoptosis. So this is annexin 5 staining with the green one, and the blue one is just the DAPI staining for the healthy nucleus. And we can see that even in 1 millimolar, there are a lot of dead cells. the cells which have undergone the apoptosis, and 10 millimolar, almost like about 60-70% cells, they died. So this proves the beta hydroxybutyrate or the ketone body is really dangerous for heart, the cardiomyocytes. Now, in one of our previous publications, we found that Pper Gamma, this is a paroxysm proliferator activator receptor gamma subunit. This is one of the molecule, one of the transcription factors, which is related with the diabetes. But nobody has done this study that whether the Pper Gamma has any connection with these mitochondrial enzymes that I have discussed about the HNGCS2 PTK4 and all. So first, we tested whether the Pper Gamma is expressed in our condition, in our system, the diabetic heart. And this is the immunohistochemistry picture. This is the chow, the regular heart. This is the cross section of the left ventricular heart. And the brown dots, I think you can see the brown dots. These brown dots means the positive staining. And you can see here the brown dots are very few, which is quite understandable since this is a regular heart. And this is the high fat diet fed heart. And you can see there are a lot of brown dots are present. So that means the Pper Gamma is overexpressed. We also did the real-time PCR to see the expression at the mRNA level, and the expression is almost double in compared to the normal. And then what we did is that Pper Gamma, we know that this is a nuclear receptor. So we studied whether the Pper Gamma is really going to the nucleus or this is some kind of artifact staining. So we isolate the nuclear fraction and the cytosolic fraction from the left ventricular heart tissue. And we blot for the Pper Gamma, and we can see in the cytosolic fraction, there is no change between the chow and high fat diet. That means the basal level is same. But in nuclear fraction, clearly the high fat diet group, the expression of the Pper Gamma is very, very high. So that means the nuclear transcription is happening and the Pper Gamma is activated in this heart. So now we actually did a little cartoon. So this is a hypothesis. After that, from this hypothesis, we actually moved on. So glucose and fatty acid, this metabolic switch actually generates a lot of free fatty acids, and Pper Gamma actually it's a receptor for free fatty acids. So when it binds to the free fatty acids, it actually translocates into the nucleus, and the nucleus there is a Pper Gamma responsive element. It binds there, and it can actually change the transcription of these genes, the HNGCS2, the BDH1, and PTK4. So this study we actually did in some isolated cell, the cardiomyocytes. It is not presented over here, but we have seen this translocation and how they actually regulate the gene expression of these genes. So if these genes are transcriptionally activated, then they will increase the ketogenesis process and beta oxidation, and that will obviously release a lot of cytochrome C and reactive oxygen species, and that causes the myocyte apoptosis and fibrosis. But this is just a cartoon. I mean, maybe it is not exactly happening like this. So that's why we moved on to study further. So next, what we did is that the Pper Gamma gene knockout. Am I running late or I still have time?
No, sir, it's okay. You can continue.
Okay, okay. Thank you. So next we did is that the Pper Gamma gene knockout. This is inducible knockout we did. So this is for the students mostly. So we use the Kreloc system. So in this system, we have an enzyme. It's called the Kre recombinase. That actually, this enzyme specifically recognize two LOXP sites. You can see this is one LOXP site and this is another LOXP site. And this enzyme, when it is activated, it actually cuts in between these two sites. So if you want to, knock down some kind of gene, you need to make sure that the LOXP sites are there. And this recombinase enzyme actually recognize this, and then it can cut this in between. So since this is a LOXP site, and this is, in English, this is called flanking, right? So flanking of LOXP, that's why it is called floxed. So in books or in literature, you may have seen this term floxing or floxed. So this is the meaning of floxed, the flanking of the LOX site. So this kind of thing, I mean, you can do a lot of things with this system. This is a very, very powerful system, this knockout system, or the Kreloc system. So this you can do to do a global knockout. That means it depends on the promoter gene you have selected. If the promoter is expressed all over the body, throughout every tissue, like ROSA26 is such a promoter which is expressed throughout the tissue. So if you choose that kind of promoter, then the knockout would be global. So that means that protein would be absent. I mean, that gene would be absent from everywhere of the body. But you can also choose an organ-specific promoter. That means, suppose you want to knock down a gene specifically in the liver. So you choose such a promoter gene which is only expressed in the liver, not in other tissues, so that you can have an organ-specific knockout. So either you can have a germline knockout. That means the animal is born without that gene, right? And sometimes that is difficult to handle that situation, because some of the genes are very important to survive. So if the gene is absent, the animal will not survive and will not grow, will not breed anyway. So in that case, there is another system which is called the Tamoxifen induced Kreloc system. Here, there is the enzyme is the Kre enzyme. Actually, it is combined with estrogen receptor and a heat shock protein, and it is present outside the nucleus. So the animal grows in a normal way. Whenever you inject a chemical substance called tamoxifen, right? So tamoxifen actually, it dissociates the complex between the estrogen receptor and the Kre recombinase, and then the Kre recombinase actually, it gets inside the nucleus, and then it does the knockout thing. So with this, at any stage of life, you can do the knockout. So this is called an inducible knockout with tamoxifen, and we use this system for our study. Now, first, we actually did some very basic experiments to see whether the Pper Gamma ablation means the knockout. So the Pper Gamma ablation or the knockout, whether it ameliorates the high fat diet mediated changes like the hyperlipidemia. So first we simply measured the triglyceride and cholesterol and all this fatty acid thing, and everything is downregulated in Pper Gamma knockout group. So from now on, this would be maintained. The white is for the wild type, right? And the black one is for the Pper Gamma flox. So this is the flox one is the knockout one. And this is the heart, the left ventricular section from the wild type chow. And this staining is called the oil redo staining, that actually stained the neutral fat which is deposited in the heart. So in high fat diet, you can see a lot of blotches of this red stains. That means the heart is full of these neutral fats. But in wild type, there are a few. In Pper Gamma knockout, even with the high fat diet, there are only a few. So that means the Pper Gamma has definitely an effect over high fat diet mediated hyperlipidemia. Then we went to the next part of the study, that whether the ablation inhibits the ketogenesis and the related enzyme expression. So what we have is that now the study becomes a little complex. So now we have 4 groups. In wild type, we have chow or the regular group and the high fat diet. And the Pper Gamma knockout, we also have the regular one and then the high fat diet one, right? So we have seen the high fat diet group, the HMG-CH2 expression is higher than the control. But with the Pper Gamma ablation, the expression actually goes down. The same thing is happening for BDH and the PDK4. So this actually signifies that Pper Gamma has a role in downregulating the HMG-CH2. The ablation of Pper Gamma actually downregulates the HMG-CH2, BDH1, and PDK4 expression. We also did immunoprecipitation reaction, where actually we have seen there's physical interaction between these proteins, but the data is not shown here. And also, we measured the beta hydroxybutyrate, that is the ketone body concentration, and we have seen a downregulation, a considerable amount of downregulation of ketone body synthesis when the animal is knocked down with Pper Gamma. So this was published recently in 2018. And then this is the same thing, but we also did the immunohistochemistry to see the, this is the microscopic picture. The same thing we can see here, that with the Pper Gamma knockdown, the HMG-CH2 expression, the protein expression is also very less in the left ventricular heart tissue. Now we did the staining. We did the staining, the regular staining, the eosin and hematoxylin staining to see the morphology, if there is any morphological changes. And then we did the picrocereus red staining on the right, which is for the fibrosis, to see the fibrosis. So in the left panel, you can see there are, so these cells are not normal. These are pycnotic cells, typical. So when the meiosis, the myocytes apoptosis happen, the cells become pycnotic. That means the nucleus is actually present at the border of the cell. And then there is also herniation of the nucleus. And then there is cellular lysis. So all these features are actually, we can see here in high-fat diet group in wild-type mouse. But when the mouse is a Pper Gamma knockout one, the change is actually ameliorated. So this signifies the Pper Gamma ablation prevents the tissue damage. And also the fibrosis, we can see a lot of fibrosis. These red fibers we can see in a high-fat diet group, which is absent both in wild type and also Pper Gamma knockout control, and also the high-fat diet one. So next important thing is the apoptosis. So sometimes what happens, maybe there is a metabolic change occurs, but it may be a transient one. That means there is reverse remodeling of the heart. So this is a very interesting topic also. People are working on that. Reverse remodeling of heart means some people with a heart failure, they survive long. Maybe after heart failure, they can survive 20, 30 years. But some people, they die quickly. They die very, like within few months. So why this is happening? Because in some people, there is reverse remodeling of the heart. The heart can actually, changes itself to make itself to normal and healthy heart, or tries to do that at least to some extent. But in some other cases, it does not happen. So this reverse remodeling is very, very important. And sometimes if the reverse remodeling happens, then whatever metabolic changes happen, it does not reflect that into apoptosis or the cellular death. So that's why to observe that if there is cellular death is happening, apoptosis of myocytes is happening, we did a tunnel assay. In tunnel assay, maybe this the resolution of the picture is not that great because this is taken from one of my papers published recently. So if you can see the green dots with the arrow, we can see the green dots. So the number of the green dots actually, it represents the apoptotic nuclei. So the number of the green dots are definitely much more in high-fat diet wild type group, but in all other groups, including the wild type control and Pper Gamma control and also Pper Gamma high-fat diet group, the number of these apoptotic nuclei are very less. And this is the quantification of the same data. You can see there is a significant change in a high-fat diet group, but with the Pper Gamma ablation, it significantly goes down. So along with that, we also did the Western blot for some of the apoptotic parameters, like I was talking about the cytochrome C and BAX and all in the last cartoon. So as you can see, the cytochrome C is expressed highly in the high-fat diet group as the BAX, and also a similar kind of expression we can see from the for the BAD. All these three are pro-apoptotic markers. So they actually facilitate the apoptosis. And with the Pper Gamma knockdown, all these markers have significantly reduced, although the quantification is not here. Now, this is a kind of mechanistic study. We wanted to see if there is really this Pper Gamma HMG-CH2, this axis, or the axis we are talking about. I mean, is it really happening? I mean, is it really important for myocyte apoptosis? Or whatever is happening, it is due to some other changes. We never know, right? Until we get to the mechanistic point. So what we did is that we did some mechanistic study with, again, the isolated cardiomyocytes. These are all primary cells. So these are not cell line. These are directly isolated from animals. So what we did is that, so this is the left corner picture. So we treated the cells with either palmitic acid or oleic acid. So palmitic acid it actually mimics the high fat diet condition, I mean hyperlipidemic condition in vitro. So this is a saturated one, and oleic acid is a control for that, because oleic acid is a unsaturated one. So we always use the oleic acid for this kind of study, just to have a control. But we'll only focus on the palmitic acid one, so only this section of the picture. So what we did is that we treated these cells either with rosiglitazone. This is a chemical compound which is an agonist of Pper Gamma. That means whenever we add rosiglitazone, supposedly the Pper Gamma should be activated in the cells. And then as another control, we treated the cells with a cathepsin G. So cathepsin G is a serine protease, which is released by the neutrophil. So whenever there is a cardiac damage, there are a lot of infiltration of the WBCs happen. For any organ, this is true, as for the heart. So these WBCs, the neutrophil fraction, they actually release cathepsin G and other serine proteases that actually damage the heart itself. So this is kind of bad for the heart, the release of the cathepsin G. So all this negative means there is no, I mean, this drug is not present, and the positive means the drug is present. So as we can see with the rosiglitazone, the Pper Gamma expression is definitely high, as well as the HNG-CS2, cytochrome C, and BAX. And the same thing, almost similar thing is happening for the cathepsin G. So the cathepsin G mediated damage is actually the same kind of damage is done by the Pper Gamma overexpression also. So just to see the mechanism with little more insight, what we did is that we did a transfection with Pper Gamma adenovirus into the cell, the same kind of cells. And we obviously have seen that Pper Gamma is expressed, as well as the HMG-CS2 is overexpressed. And these are the cardiomyocytes stained with the HMG-CS2 and cytochrome C. So this is a dual staining, and this is immunofluorescence, dual staining. HMG-CS2 is represented by the green staining, and cytochrome C is by the red staining. So as you can see, whenever only the Pper Gamma is present, or only the fatty acid or the fat is present, there is not much change. But on the absolute right picture, the third one, where both are present, that means Pper Gamma is also present. I mean, the adenovirus is present, so the Pper Gamma is overexpressed, and also the fat is there. So here you can see a lot of yellow dots. These yellow dots represent the co-localization of these two. So that means the HMG-CoA is overexpressed, as well as the cytochrome C is overexpressed. So whenever there is overexpression of the Pper Gamma, along with the lipid-rich, I mean, in lipid-rich environment, there is always apoptosis of the cells. So this is a kind of mechanistic study that actually shows that yes, the Pper Gamma HMG-CS2 axis is activated in high-fat diet condition, and that actually represents that is responsible for the myocyte apoptosis. So this paper actually was published in the Cellular Physiology and Biochemistry in 2018. And so from this study, we can conclude that the chronic high-fat diet consumption leads to hyperlipidemia, hyperglycemia, and insulin resistance. And the glucose unavailability causes the metabolic switch from the glucose to fatty acid oxidation. That is a very, very important point. And when it happens, why it happens, this thing is also not very clear. Pper Gamma induced by free fatty acids in turn transcriptionally activate the HNG-CoA2 and other ketogenic enzymes that upregulates the ketogenesis and mitochondrial oxidation, causes inflammation, apoptosis, and the cardiac damage that ultimately leads to cardiac dysfunction and diabetic cardiomyopathy. So in this study, I did not show that because the time is limited, that we also did a seahorse experiment. Seahorse experiment is a kind of ELISA experiment where you actually put all the conditions that is inside the mitochondria, and you can actually put the cells, the myocytes or whatever cells you want to have, and you can also add the lipid, whatever condition you want to have, just like inside the body. And that actually in real time, in vitro condition, that actually gives you the rate of respiration, how much ATP is produced, how much oxidative free radicals are produced, everything. So we have seen that there is a very large amount of, it's a very large change happens between the regular myocytes and this kind of, lipotoxic myocytes. So then also this molecule we studied further in type 1 diabetes, and we published this paper that HMG-CH to these molecules not only involved in type 2 diabetic heart, but also it is related with the type 1 diabetes. And this was a publication that we did in vitro, where we only focused on the cathepsin G I was talking about, the cathepsin G, which is very dangerous for heart itself if the neutrophils are overactivated. And from this, we actually did another microarray analysis, and we found multiple genes. So in a microanalysis or RNA sequencing, generally you will get thousands of genes, but there you need to set a particular cutoff value, like 1.5-fold or 2-fold changes you want to see, and then it will narrow down the number of genes, and from there you can do pathway analysis to see which genes are present in which pathway, right? Like these genes are present in cell-mediated immune system. These genes are present in cell cycle. These genes are responsible for cell death and survival, like this. So from this kind of study, we found this about 20 or 25 genes that are actually responsible for the heart disease or heart failure, especially in type 1 diabetes. I think that is enough. If the time permits, I can show a couple of very interesting slides.
Yes, sir, sure.
Okay, thank you. So this was a recent work that I did with the inducible pluripotent stem cell. So what I did is that I collected some stem cell from patients, the cardiac patient, and I was working with obviously the cardiomyopathy. So I wanted to have an isogenic control. That means, suppose if I get a patient and a healthy person, right? But these two things are actually not comparable, because healthy person is always healthy and the patient is always a patient. So you cannot actually compare them unless you have the same thing from a patient and the same thing from a control subject. So if you suppose there is a mutation in a patient, and due to that particular mutation, suppose there is lamin mutation. Lamin is a cytoskeletal protein, nuclear protein actually. So if there is a lamin mutation, there is a disease called laminopathy, and the people, they actually die at a very early age because of that. So if you want to study the genetic predisposition and all other molecules related to the disease, you want to have an isogenic control from the same patient. That means from the same patient, you want to correct that mutation so that you can have a healthy cells from that patient. And that is only possible through the stem cell. But stem cell isolation is not possible from a patient body, right? So there is a fantastic technology. It is called iPSC, or inducible pluripotent stem cell. That means any cell can be reprogrammed to produce the stem cell. I mean, literally any cell, but generally we do with the skin stem cell, and the skin cell or the keratinocytes, something like that. So what we did is that we collected skin cells from these patients, and we converted them to the iPSC with certain by expressing certain pathways and down regulating certain other pathways, and then we actually formed an artificial heart in a cell culture that actually beats. Let me show the video. I think it should work. Okay. Can you see it?
Yes, sir.
So this is the beating heart that we actually produce in a cell culture in a Petri plate, because the stem cells were fully converted to form a cardiomyocytes, and it was like a small cardiac network which is beating. So this is the 4x and this is the 10x of the same cell culture. I think that is enough. I've taken a lot of time actually. And this is the characterization of that. So these are the undifferentiated cells. Only the nuclei we can see with the DAPI staining, and when they become fully differentiated, you can see the actual shape and the typical appearance of the cytoskeleton protein inside the cell. That means these cells are fully differentiated into cardiomyocytes. Thank you.
Thank you, sir. It was really an interesting and informative session.
Thank you so much.
But we have some questions
Yeah, sure.
over here. I hope we can take it up.
Yeah, sure.
Okay. So the first question is from Shauli Day. She's saying that BAX BCL2 both can lead to apoptosis, or they are separate pathways leading to apoptosis.
Right. So what is the question?
BAX BCL2 ratio. I think she
Oh, the
wants
ratio.
to.
Okay.
I think she has not mentioned the word ratio, but I think she meant that.
So generally the BCL2 is protective, so that is actually anti-apoptotic, and BAX is the pro-apoptotic. So that's why people generally do the ratio to see if the ratio is changing. But we did not do in this study, but yeah, I mean, anyone can do that actually. So there are a lot of parameters to study the apoptosis. So some of them are BAX and BCL2, and then there is also cleaved caspase 3. So actually people do either apoptosis study, or sometimes they also do the ER stress study. So there are also a lot of ER stress markers. So sometimes what happens is that autophagy is actually upregulated and apoptosis is not, which is actually good for the heart. But in this study, we did not do that. We did not study the autophagy marker like LC kinase and all. But that is a very good way to study.
Okay, so the next question is from Faiza. She's telling that why heart attack occur more on Monday rather than on any other day.
So this is basically a Western data, frankly speaking. So people actually do a lot of partying and all, you know. So maybe the lifestyle is a major factor. Partying and all, the lot of stress, alcoholism, smoking, and all these things actually contribute to that.
Next, what prevention should be taken by the diabetes patient to prevent cardiovascular disease?
Oh yeah, this is a very important question. Very nice question. And this is very relevant actually. So for diabetes patient, so type 1 and type 2, right? I think you are talking about type 2 because type 1 is, not a lifestyle disorder. I mean, this is more like a genetic thing. And generally young age group, they actually get affected. So type 1 diabetes, obviously metformin this kind of medicines are prescribed. But for both type 1 and type 2, exercise. This is number one thing, and healthy diet. So healthy diet and exercise, these are the two most important things that you need to follow. And also there are a lot of other drugs. But again, I mean, the diabetic drugs, there is an enigma about that, because diabetic drugs are such that if you want to stop at some point of time, it may become detrimental. It may have some side effects. So it has to be continued for a long period of time, if not throughout the life. Yeah.
The next question, is there any optimal condition for CRE recombinase to function?
Optimal condition. Yes, absolutely. Because sometimes what happens, so first what we do that, we check that how much, how much recombination is there. How much, so because before doing any kind of the knockout study, first we need to check whether the system is ready for the knockout. So there is an experiment, it's called the genotyping. So first we do the genotyping before the knockout to see if all the genes or all the markers that we need. Like sometimes you also need reporter gene, like GFP, right? To see whether the promoter is there. If the cells are expressing GFP, that means, if the cells are expressing about 50 to 70% GFP at least, that means the system is good for knockout. And if the GFP expression or the reporter gene expression is very low, like 30% or less, then the system is not ready for knockout. So it depends on a lot of factors actually. First, we need to check that, because it is a very time-consuming process once you proceed with the whole thing. So before that, we need to check it.
Okay, the next question is from Pritisha Dash. What are the level of exercises that are considered to be best to prevent heart diseases?
So that depends on the age, number one, and number two, the physical condition. There are certain factors you need to consider. If the person is really aged, like a person is like 80 years age, and another person is like 50 years age. The age difference is about 30 years. So the level of exercise and the intensity of exercise will also change accordingly. But the one good thing is the yoga. Yoga is a very good thing that is actually very much doable for all the age groups, pranayama and yoga. And it is not that much hard to do. And also the motivation level is good.
Next question is from Shayantika Roy. How is heart disease related to strokes?
Heart disease is related to stroke in many ways actually. So when the clot is formed, the clot can be formed in any major artery, right? So the artery which are actually supplying the blood from the heart to the brain. So if there is a clot or if there is atherosclerosis, narrowing of the vessels occur. So there is a disease called Moa disease. So what is that? It's a very interesting thing. In this disease, heart functions perfectly well. In echocardiography, in everything, you find everything normal. But there is a particular diagnosis. It is called the carotid Doppler. So that means that Doppler is, people do from this region, from neck region, to see if there is any blockage in between the heart and the brain. So if these vessels are blocked, then there is every possibility having a brain stroke. So yes, heart is definitely related to the brain stroke.
So the next question is from Anindita Fani. Does this has any effect on gestational diabetes also?
Gestational diabetes, interesting. Generally, gestational diabetes is a transient thing, right? So it generally goes away after the birth of the baby. But sometimes it has been found that many women, pregnant women, they actually formed like kind of insulin resistance even after the birth of the baby. So in some fraction, in some percentage, the gestational diabetes, it actually continues to happen even after the birth of the baby. But how far it is related to the heart disease, I'm not sure. I cannot make any comment because I do not have any knowledge over here. But what happens is that during the gestational period, a lot of maternal changes occurs, including the hypertrophy of the heart. The heart becomes slightly hypertrophic, just like an athletic heart. Maybe not that much, but a little bit to accommodate, more blood for the baby. But these changes are most of the time transient.
So the next question is, as omega-3 fatty acid sources, we can't have the seafoods like salmon, mackerel, cod liver oil, etc. What can be the substitutions, especially the animal source for us?
Fish oil is available actually. You can find in Amazon or Flipkart. The fish oil capsules are available. You can have the capsules, and this is as good as having a fish or sea fish. If the salmon is not available here, tuna is not available here, you can have the sea fish. Even hilsa fish and this kind of sea fish also, they contain a lot of omega-3.
The next question.
Nuts also contain omega-3 fatty acids.
Next question, which factors induce cardiac fibroblasts to convert themselves into cardiac myocytes?
This is a very important, it's a very interesting question actually. To be very honest, I do not have the answer. But actually we were studying this intercommunication when I left my lab in US. Actually, we are in a project, we are studying this thing, that how they're communicated. And on the top of my mind, I can talk about a couple of factors. So there are transcription factors we all know. But recently, there are some factors discovered, known as master regulator. The master regulators are they are on the top of the transcription factor. So there are such factors, so one master regulator can actually regulate a lot of transcription factors together. So one such complex is called mediator complex. So there is mediator complex 6, the mediator complex 3 to 25, I think. So 6 and 25, these two mediator complex, they have this kind of effect over other transcription factor to change them. Actually, every cell is same, right? If you consider all the cells have same kind of genetic makeup and all, but only the transcription level and the translation level is different. So if I can reprogram the cell in such a way that the transcription and translation level becomes same, then theoretically it is easy to convert any cell to any other cell type. So mostly it is due to some master regulator.
The next question is the activation of Pper gamma related to heredity?
Related to what?
Heredity.
Yes, Dr. Shikdar, I just want to request you that if you can stop sharing the screen, because the video that
Yeah,
yes, yes, okay.
it's all right, right? Now it's all
Yes.
right?
Yes,
Yes,
sir,
sir.
it is all right.
Thank you. What was the question again, madam?
Is the activation of Pper gamma related to heredity?
Activation of Pper gamma related to heredity. So activation, so I want to clear a very small thing, but very basic thing here. So whenever we need to study some kind of heredity changes, like mutation or certain allele, I mean certain haplotype, right? We need to study the DNA. So then we need to do the PCR from DNA, the genomic DNA. But if we need to see the expression level, if the expression is changing, then we need to do the RT-PCR. First we do the RNA and then we get the mRNA population, then we do the cDNA, and then we do the RT-PCR. So the RT-PCR or the reverse transcripted PCR will give you actually the expression level, if there is any expression level changing. And the whole genomic DNA PCR will give you if there is any kind of mutation is there. So starting both the thing, like if there is any mutation in the Pper gamma, and also at the same time the Pper gamma expression is changing due to that mutation, I think it is kind of difficult to study both them together.
Okay, so next question is, could excessive use of EpiPen lead to DCM?
AP what?
Pen, P-E-N. I don't know what.
I exactly don't know what it is. If he or she can explain that, then I may try.
Chetanjana Ghosh, are you here?
I mean, she can talk in the microphone, right?
Yes, I think, so can she unmute herself?
Yeah, please. Go ahead.
What is the name, Momita?
Shwetanjana.
EpiPen.
EpiPen, E-P-I-P-E-N. It's a hyphenated word that she's written. The next question is, the gene alteration due to HFD, is it possible to actually I think it should be not transfer, down regulate from one generation to another?
One generation to another generation. No, I mean, without gene therapy, it is not possible. Not possible. In the same generation, I mean, in the same human being, it is definitely possible. That's what we do actually by giving a medicine. Suppose the energy metabolism is very less. So most of the heart disease happens due to lack of energy metabolism. So that's why doctors often prescribe the metformin. The metformin is an activator of AMP kinase that actually upregulate the whole energy metabolism. So in the same person possible, but not genetically it's not possible.
The next question is from Shubhalakshmi. If the person is having osteoarthritis, due to which she is unable to walk for more than 10 minutes, then what kind of measures should she follow to maintain her ideal body weight and not get obese?
Osteoarthritis is this is irreversible changes. Whenever you mention osteoarthritis, so irreversible changes cannot be reversed completely, but obviously, you need to take anti-inflammatory drugs, and also to reduce the weight, you need to follow the weight management thing, like the diet, obviously, and some simple exercise. You definitely.
What the next question is, like, can only diet help her to maintain the healthy weight?
Only diet is not good enough. It's a part of the whole process, but you also, continue some exercise, yoga and all.
The next question is from Dr. Shonjukta Kaul. Systolic or diastolic pressure, which one has more impact on CVD?
This is a very nice question. Very good question, actually. So let me answer in this way. So systolic pressure is the 120 millimeter, the upper one, right? And the diastolic is the bottom one, 80 millimeter of mercury. So systolic pressure, the blood is actually pumping out from the heart and going to the body. So even if the pressure increases a little bit than normal, it actually increases whenever you do some exercise. Even when I talk, right, my systolic pressure will automatically increase. So this kind of increase does not cause any harm in general because the pressure is distributed to the body. So this is kind of divergent thing. But the opposite is the convergent thing. Diastolic pressure, the blood is coming to the heart. So if that pressure increases too much, that directly causes some kind of cardiac problem. So yes, I mean, for heart, the diastolic pressure is obviously more important. It's more pathological if it increases.
Okay. The next question from Shamapti. How many blockage of cardiac valves lead to first time heart attack?
I don't know actually. So this is a question for the clinician.
Okay, so the question from Shwetanjana that she wrote was epinephrine. Actually, it should be epinephrine.
Epinephrine.
Could excessive use of epinephrine lead to DCM?
Yeah, I mean, it can indirectly because all the sympathetic neurotransmitter, they actually increases the heart rate. So if you continue to have that with some kind of artificial means, yes, it can do that actually. Because if the heart pumps a lot, I mean, with more force for a longer period of time, definitely there will be a possibility of dilation. Yes.
I think there is no more question.
Momita?
Yes.
Madam, I have got a question through the WhatsApp. One of our students has sent it. The name of the student is Deepshika. She has asked that we all know that HDL is the good cholesterol and LDL is the bad cholesterol.
Right.
So why HDL do not get stored into the artery, whereas LDL can get stored?
HDL?
HDL do not get stored into artery. It cannot get deposited into the artery. I think she is asking.
So what happens is that LDL does not store directly into the artery. So LDL is actually, it sends a signal like an antigen. It has, I mean, it sends an antigenic signal, and the circulating macrophage, they actually consider it as an antigen, and with the endocytosis, it actually engulfs those LDL cholesterol, and that forms the foam cells, the huge foam cells actually, and those foam cells, they actually lost their ability to move further. So whenever they get some space during the transportation, during the circulation, which is ideally the the arterial bed, so they tend to deposit in the wall. So it is not the LDL cholesterol, it's the foam cells.
Okay, sir. I have got one question, and that is, like you said, that there is a switch over of energy metabolism, like from the glucose, the heart shift towards the fatty acids. Like for those who are practicing the keto diet, is it possible for them also?
It is possible. So actually, sudden change of any diet is bad. That is my concept, my understanding. So suppose a person overnight he has decided that I'm kind of overweight. So from tomorrow I'm gonna completely change my diet and I'll go to the keto diet. So no carbohydrate, only protein and fat, ghee and oil on this thing. So this kind of sudden change in diet is not good because your body is not ready. Number one, metabolically is not ready. That is the number one point. And second point is, are you doing enough exercise to metabolize that? Are you doing enough exercise? If you're only sitting home, just watching TV and just changing your diet, that's not going to help actually. So changing diet suddenly, it's not a good thing. Accordingly, according to your exercise level, your daily activity, you need to change your diet.
Okay, thank you, sir. It was a wonderful session. Now, I would like to request our head of the department, Dr. Shamsubha Thakur, to give a vote of thanks.
Thank you, Dr. Dash. First, I want to thank Dr. Shikdar for this wonderful talk. Really, it was a wonderful talk. Dr. Shikdar has started from the very basic, which is very required for all the students, and thereafter he has given the details of his work that is going on right now. So it was a very and I think that from the number of questions that is coming up from the students, it only signifies how much the students have enjoyed the talk. And the students have really enjoyed the talk. And I think they have learned many things from this talk.
Thank
So
you.
thank you, Dr. Shikdar. And I want to request you that in this situation it is difficult, but whenever the situation will be in the neo-normal situation, we will all from the department, we will all like to invite you in our campus, so that our students can directly interact with you, and I think the students will like to do that. I also want to thank Dr. Nilanjan Das for his
My email ID, if they have any questions, so my email ID they can note down and they can send the questions. So my email ID is kunal.shikdar, KUNAL. I can actually write down in the chat box. So if they have any question, they can always email me.
Thank you, sir. I also want to thank Nilanjan sir for his support for carry out this whole webinar series, especially whenever he said that I want to request Dr. Shikdar, as I had a previous interaction with Dr. Shikdar regarding some other issues, if you can remember, sir. So I said we should have him in this webinar series. Thank you, Momita ma'am, for your fine moderation, your wholehearted effort for the webinar series. I want to thank all the students for participating in this webinar series. I also want to thank Wendrila ma'am and Ina ma'am for their help in conducting this webinar series. It will go on for next 6 days. There are 6 more sessions. So I think all the students will attend, and I think they will all be able to interact with some eminent persons like in the previous 2 sessions. So with all this, I want to hand it over to Momita.
Yeah, thank you for this session. Today it's over for the day. Tomorrow again we will meet at the same time, 7 p.m. with Professor Sanjeev.
Momita, just for a second. Momita,
Yes.
some of the participants are saying that the
Nilanjan sir.
that
Yes, sir. Your voice is cracking, sir.
somebody saying that it's not working.
What is not working?
Feedback form.
Okay, feedback form. It will open after the session, sir. It will open after the session.
Yeah, just
gets over.
announce that. Just announce that.
Yes, sir. I've written it down in the chat box that the feedback link will open after the session is over.
Okay.
Okay, sir. Thank you, and hope we'll again meet tomorrow with Professor Sanjeev as our speaker for tomorrow from 7 p.m.
Thank you so much. Have a good night.
Good night.
Thank you, sir.